Welcome back to the third and final video in our series discussing VDJ recombination and an overall module thinking about the genetics of how we put together antigen receptors. And so in the first two videos in this series, we mostly focused on somatic recombination, VDJ recombination as it related to the construction of immunoglobulin molecules. In this video, we are going to now turn our attention to the T cell receptor, which follows mostly the same set of rules, although we'll discuss just a few areas of divergence, as well as just some sort of special considerations about the topic of VDJ recombination as it applies to the overall repertoire of both immunoglobulin and T cell receptors. But let's start just kind of with the nuts and bolts of how we put together the T cell receptor. So recall that conventional T cell receptors have an alpha and a beta chain. There are such a thing as gamma delta T cells as well, but we're not worried about them for right now. We're going to focus on old-fashioned alpha beta T cells for this lecture. And so there we have an alpha chain and a beta chain, and both of those chains have a variable region and a constant region, and it is the variable region that interacts with antigen, as we've seen before. And to put together the variable alpha region, we are going to need V and J segments. And so this means that the alpha chain of the TCR is analogous to the light chain of the immunoglobulin, at least in terms of VDJ recombination. So just like the light chain, the alpha chain locus contains V and J segments, but no D segments. We only have V and J here. And you can see that there are actually quite a lot of both of these segments in comparison to the IG light chain loci. We have, somewhere between 70 and 80 V alpha segments, depending on the individual, and again, around 61 potential J alpha segments. And we can recombine one of these Vs with one of these Js. And to think about the overall combinatorial diversity here, you would multiply, again, this number by this number, which would add up to be, several hundred at least. And you can see that on the final protein that is made, that both the V segment and the J segment end up contributing to the final the protein structure, and again, most importantly, to this most N-terminal region that is going to interact with antigen, but not only with antigen, because now we're talking about the T cell receptor, it also needs to interact with MHC. So the folding or the combination of VD and J segments yields a protein that is capable of both interacting with MHC as well as a peptide antigen that's being presented by that MHC. Having been introduced to the segments in the alpha chain locus, V and J segments, we can look at how they are put together at the different levels of the central dogma, going from DNA to protein. We can see that there are different numbers of V segments, again, depending on the individual, but there are many. There's some number of J alpha segments as well. And again, we pick one V, we pick one J randomly, and we recombine them together, and all of the intervening DNA will be cut out, just like we saw with immunoglobulins. Segments that were not cut out remain in the DNA, of course, but they do not end up coding for anything. So they are left behind, but they do not themselves form functional exons that will encode for anything, and they will eventually be spliced out at the RNA level, which is what you see here. This recombined V alpha, J alpha exon is kept following splicing and actually encodes for the TCR alpha chain that we make, along with the constant alpha segment. Again, this is a conserved sequence in the germline and is not recombined in any way because it encodes the structural constant region of the final alpha chain. So that's how we use recombination to put together a TCR alpha chain. Should look familiar if you watched the previous videos about immunoglobulin chain recombination. So let's follow this logic then and introduce ourselves to the beta chain locus. So the beta chain locus is more analogous to the immunoglobulin heavy chain locus because it has V, D, and J segments. It has all three. But notice the structure of this locus is slightly different. So there's one kind of caveat here to the beta chain locus if we think about it in comparison to the heavy chain locus. We still have quite a few V segments which we can use. There's actually usually only two D beta segments. And each of these D segments has its own set of J segments that it can recombine with. And if, for example, the cell ends up using D beta 1, it will only use this first set of 6 J segments to recombine with. And it'll use a constant segment known as constant beta 1 because it is immediately downstream. In contrast, if it uses D beta 2, it will only use these downstream J segments. Because remember, you can only recombine with segments that are downstream. So because these J's are upstream of this D, that means that that this D cannot recombine with them. It can only recombine with its own downstream J's. Conversely, this D will only recombine with this set of J's because there's just a little bit too much DNA in between, including this constant region. And so recombination happens very inefficiently if RAG tries to reach this far down the locus to reach these J's. So the functional consequence of that is that you actually do not have the full set of combinatorial possibilities here. Because each D is restricted to a subset of J's, the combinatorial diversity of the beta chain locus is limited somewhat. So the math that we did before would work a little bit differently here, but we won't go through it. In any event, whether or not you use this cluster of D and J or this cluster of D and J, ultimately you'll put them together and you'll combine that with a V. And again, that will form the variable region of the beta chain that will interact directly with antigen and with MHC. And if we look at the way these events come together, you can see this here. You would have our number, whatever number of V, D, and J segments in the germline DNA. This is not pictured here, but I'll tell you that just like in the heavy chain locus, the D and the J segment always recombine first. And we'll look at one mechanism that enforces that sequential recombination. But for right now, just take my word for it that D and J recombine first, and then whichever D and J combination that we use will recombine with a V segment, which will form a functional exon, as we've seen, which will be included in a final processed mRNA molecule, which includes the constant segment, and that will encode for this final protein, which includes all 3 VDJ segments again, with the VDJ being most determinative in terms of the function of the N-terminus of the beta chain, which will interact with MHC and with antigen. So this should look familiar. It really mostly follows the same rules. We saw just a little bit of difference in terms of the way the beta chain is structured. But overall, the alpha chain looked like recombination in the immunoglobulin light chains, and the TCR beta chain looks like recombination as it occurs in the immunoglobulin heavy chain. Another similarity is that the TCR VDJ segments also have to follow the 23-12 rule in order to recombine. So we still use RAG to recombine them, and RAG still needs to bind to one segment with a 23 base pair spacer and another segment with a 12 base pair spacer. A key difference here is that compared to the heavy chain, the D segments in the TCR beta chain kind of have one of each spacer. That is just a little bit of a trivia fact, but it is the case here. And this also just, again, enforces the recombination sort of happening on the right side. So the D segment will only recombine in this direction with the J segment because they have matching spacers and so on the other side, or complementary spacers, I should say. But how is it in the beta chain that I said that we only recombine D with J first, or we always recombine D with J first prior to recombining D with V? And also, why is it that we don't recombine V directly with J? Theoretically, that might be possible because the V segments have 23 base pair spacers and the J segments actually have 12 base pair spacers. So they are theoretically complementary to each other. Well, the reason that doesn't happen and that we always start with D recombining with J before V is ever involved is that the V segments end up being sequestered in what we call inactive chromatin during the initial recombination of D and J segments. So remember that, again, we draw DNA typically in these diagrams just as a nice little straight line, but that's not how DNA really exists. And even in terms of its double helix, it's not just kind of like one long strand of double helix that's just floating around. DNA is actually quite compact. and it exists in structures called chromatin, where it's usually condensed and oftentimes kind of folded or wound around structures called nucleosomes. And when DNA is tightly wound up in these chromatin structures, that means that it can't effectively be opened, and enzymes like DNA polymerase or RNA polymerase really don't have access to the strands of the DNA molecules in order to facilitate whatever it is that their function is. So when a T cell is developing and needs to make a TCR, what happens is that the chromatin opens up such that only the portion of the beta chain locus that contains the D and the J segments becomes loose and accessible, whereas the portion that contains the V segments remains tightly wound around the nucleosome. And it is only after the D and the J segments successfully recombine that the cell sort of receives a signal that causes the portion of DNA with the V segments to relax and sort of provide access to the V segments. But by the time that happens, D has already recombined with J, and it's then ready to recombine with the V. So this enforces, again, the sequential nature of recombination in the locus. D always recombines with J first, but this also prevents V to J recombination from occurring directly because J is sort of already taken up by D before it would ever have any access to V segments. So this is one way that chromatin regulation is important for VDJ recombination, specifically for TCRs. Okay, so another special consideration that I want to put into your mind now is we've been talking about these recombination events, and we've been kind of looking at the DNA as sort of a single entity. But remember that for all genes, and by extension for all gene segments, we always have two copies of those genes. We have two alleles. We have one set of genes on the maternally inherited chromosome and an identical, or maybe not always identical, but a matching set of genes on the paternally derived chromosome. And that is also the case for all these V, D, and J segments. All the V beta segments, for example, have their counterpart on the other chromosome. So you have maternal V segments and paternal V segments. So theoretically, we could perform somatic recombination on both chromosomes and therefore theoretically come up with two unique heavy chains, two unique light chains, two unique beta chains, two unique alpha chains, depending on the cell type that we're in. But that is not what occurs in practice most of the time. And so let's just talk about what regulates that. And that is a concept that we refer to as allelic exclusion. In a nutshell, allelic exclusion means that we end up expressing a protein using only the genes found on a single allele on one chromosome, and we inactivate or exclude the matching set of genes on the other chromosome, the chromosome derived from the other biological parent. And so if we look at that in the context, we'll start with immunoglobulin. A note that I haven't said yet is that when we want to recombine to make an immunoglobulin, we always start with recombination of the heavy chain. So recombination happens in the heavy chain first, and the cell, basically the B cell, the developing B cell, will sort of randomly choose, if you will, one chromosome or the other to attempt recombination on. And it's a random stochastic process. So on either the maternal or the paternal chromosome, we'll start trying to recombine a heavy chain. And remember that that's successful, only about 33% of the time. That's an oversimplification of things, but remember that a lot of times you end up causing mutations and it goes wrong, but you actually get more than one shot at it. And one reason that you get more than one chance to recombine the heavy chain is that you can try on both chromosomes. So if it's unsuccessful on the first chromosome that you try it on, you can then try again on the other chromosome, and hopefully between the two chromosomes, the B cell can make a functional heavy chain. And assuming that it does, what will happen is that the chromosome on which the heavy chain is successfully recombined, that heavy chain, that recombined genetic material will be expressed, but the matching segments on the other chromosome will be inactivated or suppressed. So we call this allelic exclusion of the other heavy chain locus. If you're lucky enough to successfully recombine a heavy chain on the first attempt, on the first chromosome that you go for it on, and that is successful, that will similarly transmit a signal that will allelically exclude the other chromosome before recombination even happens. So the consequence of this is that the B cell will only ever have one type of heavy chain because it will only use the genetic material on one chromosome or the other, and it will actively suppress and exclude, again, through chromatin condensation doesn't sound right, but it will condense the chromatin and render those genes inaccessible on the chromosome that it's not going to use. And that is the process of allelic exclusion. Okay, and I should say that if neither heavy chain locus successfully recombines, the B cell just dies, because without a functional heavy chain, there's really, again, not much point to it. But assuming that we did recombine one or the other successfully, we then will proceed to start to try to recombine a light chain to combine with that heavy chain. And B cells will always attempt to make their light chain using the kappa locus first. So I said before that there are two loci that encode light chain, and we can use one or the other. The B cell, for various reasons, will preferentially start with trying to recombine one of the chromosome or one of the kappa loci on either the paternal or the maternal chromosome. Which chromosome it uses is random. But ultimately the same process plays out, where if that first recombination event at one of the kappa locuses or loci is unsuccessful, it will try again on the other chromosome's kappa locus. And if neither kappa locus successfully recombines, then the cell will move on and start then to try to recombine on the lambda loci. And again, it will get sort of two shots because it'll try first on one, either the maternal or the paternal, and if it's unsuccessful yet again, it will finally try on that fourth chromosome, whichever the final lambda locus that it hasn't tried yet. But assuming that any of these four chromosomes recombine successfully, no matter which one recombines successfully, the other three will be allelically excluded. We will turn off expression from all three of the other chromosomes. So if a kappa light chain successfully recombines on one chromosome, then the other kappa locus will be suppressed, and both lambda loci will be suppressed because we only want one light chain, and it's only going to come from one, in this example, one kappa locus. Conversely, if both of the kappa loci fail to recombine, but one of the lambdas does successfully recombine, then that means that the other one of the lambda loci will be suppressed, and both of the kappa loci will be suppressed. So again, we only want one light chain. It can only be kappa or lambda, and so whichever one we successfully recombine, out of our four possibilities, we will suppress the other three, and that's what these text boxes sort of lay out for you. Okay, so T cells follow similar rules to this, although there is one big exception, as we'll see in just a second. But just like whereas in B cells the immunoglobulin heavy chain recombines first, in T cells the TCR beta chain begins to recombine first, and it's a very similar logic. Looking on chromosome 7, where the beta locus is, we will randomly start to try to recombine the VDJ segments in one of the beta loci, and if we're successful, we will allelically exclude the other one. If we're unsuccessful, then we will move on to the second chromosome, the second locus, and try there. And again, as long as we successfully recombine one or the other, we will exclude the one that we are not going to use. And in T cells, successful recombination of a beta chain transmits a signal which induces recombination in the alpha chain. So you have to recombine the beta chain first before you start trying to recombine alpha chains. This is where we find the exception to the rule that we've been building up here. Unlike all of the other antigen receptor loci, the alpha chain loci, for whatever reason, they start to recombine simultaneously. So they don't sort of go one at a time. You start recombining in both loci, and hopefully one or the other recombines successfully. But of course, if you have, again, about a 1/3 shot on both chromosomes, that means that if you multiply 1/3 times 1/3, about 1/9 of the times, around 11% of the time, let's say 10% of the time, you will actually successfully recombine on both chromosomes. And that means that it is theoretically possible for a TCR actually to make two unique alpha chains because we do not observe allelic exclusion on the alpha chain. So this is a theoretical possibility, and what that means is that these T cells, about 10% of T cells, more or less, could theoretically recognize two unique antigens. They're probably going to be similar antigens because there will always be a shared beta chain. There's always going to be a single beta chain, but that beta chain might pair with two unique alpha chains, giving you two technically distinct TCRs. In practice, however, usually one of the alleles ends up dominating for various reasons. Typically, one of the alpha chains just shows a much higher affinity for antigen than the other one does. And so the T cell ends up sort of using it preferentially. But again, it nevertheless is the case that T cells break the rule a little bit and that they might recognize sort of two structurally similar but distinct antigens, although that is rare. Okay, so we've sort of covered some special genetic considerations there. I want to also talk about a little bit another way that the TCR is distinct from immunoglobulin in terms of diversity, because as we saw in a previous video in this series, there's actually more overall diversity in the TCR repertoire than there is in the immunoglobulin repertoire. We saw that there are 10 to the 15th at least possible TCRs. Well, there's only 10 to the 13th possible immunoglobulins. So where do the other two orders of magnitude come from? Well, if we simplify a bit, we can mostly attribute the extra diversity in the TCR repertoire to extra junctional diversity. Um, T cells also have a little bit of, also have a higher degree of combinatorial diversity because they typically have more VD and J segments overall. But by far the bigger contributor to the extra diversity in the T cell repertoire is junctional diversity. And the primary explanation for that is the differential expression of the enzyme TDT, which occurs during the recombination events that make up the various chains of the TCR and the immunoglobulin molecules. So through evolution, as it turns out, the expression of TDT is absolutely the highest in T cells while they are trying to recombine their TCR beta chain. And because TDT expression is extraordinarily high, that means that there's just a lot more TDT around, and therefore more N nucleotides are going to be added on average between every junction, meaning between every DJ and then between every V and D. And 10 to 20 nucleotides means that you're going to add, somewhere like between 3 and 7 amino acids in between those junctions. That's going to create quite a bit of new diversity, new sequence diversity in the final protein product. Now, in comparison, the TDT expression during the recombination of the heavy chain, which again is kind of analogous to the TCR beta chain, it is high, but it's relatively moderate compared to the expression during TCR beta recombination. And because we have TDT expression during the heavy chain recombination, but it's not quite as high, that means that on average we add fewer overall N nucleotides between each junction in the heavy chain. And that means that there's going to be fewer amino acids that are introduced during successful recombinations. And so that limits the overall possible junctional diversity that can be produced through the activity of TDT. Another point here is that actually TDT expression is quite low during recombination of both the TCR alpha chain as well as the two immunoglobulin light chain loci. And in fact, recombination of either the TCR beta chain or the immunoglobulin heavy chain is actually a signal that turns TDT off. And so TDT expression is usually sort of just residual or left over during the recombination of TCR alpha and IG light chains. And so for that reason, we actually end up adding very few N nucleotides to these two chains. And so junctional diversity is relatively limited in the TCR alpha and immunoglobulin light chain locus. And so all of that extra diversity that gets added is much more sort of incorporated, again, to a high degree in the TCR beta chain and then to a moderate degree in the immunoglobulin heavy chain. Okay, so one final point that I want to make here as we think about junctional diversity and how it is unique is sort of to ask the question, well, why is it that T cells in particular have so much higher junctional diversity? And why do they have higher TDT expression? Why do they rely on this mechanism to confer extra diversity that immunoglobulins don't seem to need? Um, there's a structural explanation for that, and that is that the diversity that is added through junctional diversification, meaning the addition of N nucleotides between the junctions. Remember that these nucleotides are being added in the spaces between the V and the D segment, as well as between the D and the J segments. These junctions are incorporated into a single hypervariable region on the antigen receptor. This is true both for immunoglobulin and TCRs. This picture only shows TCRs, but this is true for both. Um, the hypervariable region that is constituted by the junctions is always hypervariable region 3 or complementarity determining region 3, CDR3. Remember that the CDRs are the most N-terminal sequences, the most N-terminal kind of loops in the structure of the protein that touch antigen directly. And so the diversity within these regions is what is most important and most critical for determining the complementarity of the protein, meaning which antigen, which unique antigen is going to be capable of binding. Um, and while in immunoglobulins, all three complementarity determining regions touch antigen directly, in T cells, that's actually not quite true because remember that the substrate of the TCR is not only antigen, it's the combined shape of MHC and antigen. And if you kind of look here, you can see that the yellow and the green associated with the D and the J segments are kind of restricted to like this inner portion, this kind of medial portion of both the alpha and the beta chain. And this is actually where the peptide binds to the TCR, whereas CDRs 1 and 2, they actually interact more directly with the MHC molecule. So given that CDRs 1 and 2, we need them and they are important for binding to MHC, and so it's important that they still have sequence diversity. But it's only CDR3 that binds directly to peptide antigen itself, directly to the antigen that's being presented. And so that has created an evolutionary selection pressure wherein the addition of all those N nucleotides, which again are going to be concentrated within CDR3, that is going to enhance the number of peptides and unique antigens then that the TCR is going to be able to recognize because that interaction happens uniquely and specifically at CDR3. So another difference in the way that TCRs function, in a way that junctional diversity is uniquely and especially important for binding of TCR to antigen, but also for putting together the overall diversity of the TCR repertoire. Okay, let's summarize this final video about VDJ recombination. We saw that TCRs, just like immunoglobulins, also use V, D, and J segments. Alpha chains were analogous to the immunoglobulin light chains in that they use V and J, whereas the TCR beta chains were analogous to the heavy chains of the immunoglobulin, and they use all three V, D, and J segments. And we saw that TCR gene recombination is analogous to the processes that happen for immunoglobulins. We still use the RAG recombinase enzyme, and it still has to follow the 2312 or 1223, however you prefer, that rule in terms of recognizing recombination signal sequences. And just like for immunoglobulins, all the same processes play out wherein we add P and N nucleotides to provide junctional diversity. However, overall, TCRs have more theoretical diversity, and that is mostly attributable to the higher junctional diversity in complementarity determining region 3 of the TCR, and that arises from the relatively higher expression of TDT during TCR beta chain recombination. And that gives us a really staggering number of possible TCRs that we can make, at least 10 to the 15th. But a point that will become important later is that even though we can make so many TCRs, we don't keep them all around, and especially we don't keep the ones that recognize self. So many of those 10 to the 15th 15th possibilities, are going to be TCRs that recognize self-antigens, molecules associated with the host. And so in the process of T cell development, we want to delete any T cells that are self-reactive. And so we'll see how that plays out later in the course. Okay, that covers it for VDJ recombination. In this module, we'll have one additional video where we talk about a different type of somatic recombination, which has nothing to do with the diversity of the antigen binding repertoire, but instead determines the constant region of the heavy chain of immunoglobulin, and that process is referred to as class switch recombination. I'll see you in that video.